and Lorenzo, his group applies the toolkit of nucleic acid nanotechnology to design advanced biomimetic systems applicable as biophysical models or tackling challenges in biomedicine. Okay, so without further ado, I now give the floor to Lorenzo, and looking forward to your lecture.
Thanks a lot, Martin, for the kind introduction. Thanks everyone for attending today. And as so as Martin anticipated, we're gonna go through a very, let's say, basic in a way introduction to nucleic acid nanotechnology. So that's DNA and RNA nanotechnology, and we're going to see a little bit how this can be used to engineer synthetic condensates, which of course is one of the key topics of our doctoral network. I'm not going to go into too many details for that because otherwise it would take 3 hours. Hopefully there will be another lecture in the future in which we really look at the details of how these nucleic acid condensates can be engineered. So let's say the objective of today's lecture is really giving a fundamental understanding of the principles of nucleic acid nanotechnology, and hopefully our students and others can then apply some of these principles to their own research or do further research to kind of look a bit more closer, learn a bit more about this, I think, quite fascinating topic. So who invented, let's say, DNA nanotechnology? In this case, is one of those situations in which we can answer exactly that question. So DNA nanotechnology is widely acknowledged as having been invented or introduced by Professor Ned Seaman, who sadly passed away only a couple of years ago. Was professor at NYU. And we can pinpoint pretty much exactly the origin of this research field in this 1982 paper by Ned Seaman in the Journal of Theoretical Biology, where Ned wrote that it appears to be possible to generate covalently joined three-dimensional networks of nucleic acids, which are periodic in connectivity and perhaps in space. And these are some of the diagrams in this original paper. So this was, in a way, the revolutionary statement. And what this statement really introduced is the concept of building things, building materials with nucleic acids, because of course, up to that point, what people consider nucleic acids, which is still mostly true, is as considering them as biological entities that carry and propagate genetic information, not as building materials. So that's the really groundbreaking concept that was introduced in this paper. And since then, the field has grown massively. There are now hundreds, probably thousands of research groups working with nucleic acid nanotechnology one way or the other. And we went from this very, very simple nanostructure that we're going to look at in some more detail that were, again, theorized in that paper by Ned Seaman, all the way to kind of very complicated and advanced nanostructures and materials, from tiles to DNA origami to scissors to ginormous assemblies of many origami, all the way to DNA bunnies. And hopefully today we'll understand how all of that is possible. So if you want a definition of what DNA nanotechnology is, that's one, which I guess is as good as a few others, and that's that DNA nanotechnology is a collection of tools and methods that include experimental methods, theoretical methods, computational methods that uses the physical and chemical properties of DNA and RNA. So we're going to touch on RNA nanotechnology very, very briefly today. But quite often when people talk about DNA nanotechnology, they really also mean RNA. And you want to use these tools to control structure and dynamics at the nanoscale. And this has many applications in fundamental research, and some of you will be working on these topics, but also let's say beyond fundamental research to healthcare, sensing, materials, microscopy, and controlling biological systems. So traditionally, the field has been divided in structural and dynamic DNA nanotechnology. That's a little bit of a blurred interface, or a very blurred interface, but we'll maintain this division for this lecture. So that's an outline of the lecture. So first, we're going to spend a little bit of time, quite a substantial fraction of the lecture, to review the structural, thermodynamic, and kinetic properties of DNA. because that's really, really important if you want to understand why DNA is an excellent material for constructing things at the nanoscale. Then we're going to see how you do that. So how you construct materials at the nanoscale using DNA. So there will be a structural DNA nanotechnology. And here we're going to touch on how you make condensates out of DNA, again, relevant for our doctoral network. Then we're going to look at a, say, very big subtopic in DNA nanotechnology, which is DNA origami, that I'm sure you have heard about many times, and it's so big and successful and prominent that it deserves its own little section. It's not going to be a long one. Then we're going to look at how we control the dynamics of these DNA materials and nanostructures. And finally, we're going to touch on RNA nanotechnology and RNA condensates. So let's get started. So I'm not entirely sure about what the backgrounds of the various people in this call are, but I think probably many of us here will be a vague idea of what DNA looks like from a chemical point of view. I'm not a chemist, so probably I have a less good idea than many of you here, but there's not much that we need to understand if we want to do DNA nanotechnology. So of course, DNA is a biopolymer that has a backbone, which is made of alternating phosphate groups and sugar molecules. Of course, it's deoxyribose. And these phosphate groups are negatively charged under physiological conditions, which is important. It means that DNA molecules repel each other. It means that you need cations, you need some salt in order to screen these negative charges and allow DNA to interact with other DNA. And then, of course, to the sugar molecule, you have some nucleobases connected. And in DNA, you can have 4 of these. You can have cytosine, you can have thymine, you can have adenine, and you can have guanine. And these are shown here. They're divided in purines and pyrimidines, where the pyrimidines are the smaller ones and the purines are the bigger ones, A's and G's. The key characteristic of these nucleobases is that they base pair. Again, you've learned this in probably high school biology. The A will base pair with the T and the G will base pair with the C. And this base pairing is mediated by hydrogen bonding. You can see here, C and G form 3 hydrogen bonds, 1, 2, and 3, and A and T form 2 hydrogen bonds. So CG bonds are a little bit stronger than AT bonds. But still, these are not irreversible. They are hydrogen bonds, they are physical interactions. That means that they are, to some extent, thermally reversible. You don't need to heat up DNA too much for these interactions to fall apart. That's, of course, super important in biology. You don't want DNA to be glued to other DNA in an irreversible way, because DNA needs to be read, copied, manipulated by enzymes in our cells. But that's, of course, also very, very important for self-assembly and nanotechnology. DNA forms double helices. So if you have 2 DNA molecules, that they are the reverse complementary of each other, so that's important. DNA has to be complementary to bind to other DNA, but it has to be the reverse complementary. We can go back actually and see it here. So DNA has a directionality. You can define a direction along your DNA chain, and for 2 DNA molecules to bind to each other through base pairing, they need to be oriented in an anti-parallel way. So when 2 DNA molecules are the reverse complementary of each other, means that they match if you kind of put them in an anti-parallel direction, then they will form a double helix. The process is called hybridization. So now DNA can form many double helices. The famous one that you've seen many times and heard about is actually called bDNA. That's because it was observed after the A version. So that's about 2 nanometers in width, and you need to take about 10.5 steps up your DNA ladder to do 1 full turn around the DNA double helix. It's also slightly asymmetric. It has major and minor grooves. So if you look at this ladder, from one side, you have a little bit more space in between your 2 DNA strands. Then you have on the other side, that's your major groove. And on the other side, you've got the minor groove. So that's the bDNA, is the one that is formed more often under physiological conditions, is the one that's formed more often than not inside the cells. There's other structures that DNA can form. For example, it can form an aDNA helix, so that's a little bit more compressed and chubby. DNA can form this under certain non-physiological conditions, but this is quite important because RNA molecules that can also hybridize to other RNA molecules will actually form an A-type double helix. DNA can also form this other type of helix called the zDNA. So A and B are right-handed helices. This zDNA is actually a left-handed helix that can be formed if you twist the DNA the wrong way around. It's not super important in nanotechnology. So let's have a look at the thermodynamics of DNA binding and unbinding. Again, that's super important if you want to understand the basics of DNA nanotech. So it's quite complicated to describe thermodynamically how long DNA molecules interact with each other, but it's not that complicated to describe when short DNA molecules interact with each other. And that's the kind of DNA molecules that we use most often in DNA nanotechnology, say, I don't know, up to 50, 60 nucleotides, maybe a bit more as well, and we call these oligonucleotides. So if you have two such short DNA molecules, call them A and B, assume that they are the reverse complementary of each other, so they can hybridize to form this AB molecule. The assumption that you can make is that, you can write it down as a chemical equation here, is that A and B are either bound and fully bound, or they are fully unbound. You assume that all of the partially bound states in which A and B are kind of partially connected to each other, they don't last very long. This is called the two-state model, where the two states are either the things are bound or the things are unbound. Under this very simple model, you can write down this as a chemical equation. You can write down an equilibrium constant for this chemical equation, and you can link the ratio of the concentration of the AB duplex and the individual A and B strands to a free energy of hybridization through an exponential factor. And this free energy of hybridization, of course, is a Gibbs free energy. We call it the standard Gibbs free energy of DNA hybridization. What does the little zero means is the standard one defined under some standard conditions, which typically means a concentration of the strands equal to 1 molar, but other conventional concentration references can be sometimes used. So that's this delta G0, and then of course R is the ideal gas constant, and T is the temperature. You can split your hybridization free energy in an enthalpic term and an entropic term, with it being a Gibbs free energy. So you got your delta H0, which is the standard hybridization enthalpy. So this is due to the forces that keep together your DNA double helix. So we mentioned hydrogen bonding, so that will be accounted by this term here, but you also have other important interactions, and perhaps even more important than hydrogen bonding, it's stacking interactions. So the nucleobases, they have pi stacking, they stack on top of each other, and that stabilizes the double helix quite strongly. And these stacking interactions would also be captured by this delta H parameter. The delta S is the standard hybridization entropy. So that's the change in entropy when you go from two, let's say, non-bound, non-hybridized DNA molecules to the DNA duplex. There's a change in entropy. There's actually a decrease in entropy of the system, because you have these two DNA molecules, and the DNA molecules, when they are single-stranded, when they are not in a duplex, they are actually quite flexible, and they have a lot of conformational freedom. When they bind to each other to form the duplex, they lose a lot of this conformational freedom, and you pay this in terms of a decrease in the entropy of the system. So delta H is negative, also delta S is negative, but because you've got a minus sign in front of the entropic term in your Gibbs free energy, of course, these two contributions oppose each other. So the change in entropy pushes against the formation of duplex, and the change in enthalpy pushes in favor of the formation of the DNA duplex. Now, it also turns out that delta H and delta S are, to a good approximation, independent on temperature. So you can assume that all of the temperature dependence in this delta G is in this T term that you have here. So what this means is that if you change the temperature, you can modulate the relative weight of the entropic term and the enthalpic term. And what you have, the result of this, is that DNA will actually fall apart as you increase the temperature. DNA will melt. Okay? So here is because at high temperature, obviously the entropic term will dominate over the enthalpic term, and the entropic term pushes for pulling apart your DNA molecules. So DNA will melt. This is an experimentally obtained, which I think I obtained during my PhD. It's very old, melting curve of DNA. where on the vertical axis, you have the fraction of DNA molecules that are hybridized. So these are two short complementary DNA molecules. And the horizontal axis is just temperature. And you can see at the low temperature, you have 100% of the molecules being hybridized. At high temperature, you have 0% of the molecules being hybridized. And in between, you have this nice, smooth sigmoidal trend. You can define a temperature at which the probability of hybridization is 50%, and that's the melting temperature of your DNA. It's a very simple exercise to try to work this out from the expression of the free energy and the expression for the equilibrium constant. And you should try to do this at home and see if you get the same answer for how to express the melting temperature as a function of delta H0, delta S0, and C being the concentration of the individual molecules. And this is true under the assumption that the molecule A and the molecule B initially have the same concentration. An important question, which is actually very fundamental to DNA nanotechnology, is can we actually predict the free energy of hybridization of DNA molecules based on the sequence of the DNA? That would be very nice, very useful, because it means that then we can program the thermodynamics of our DNA constructs. And it turns out that you can do this, and you can do this thanks to a very useful model called the nearest neighbor model for nucleic acid thermodynamics. So what this model basically tells us is that if you have two DNA molecules, again, call them A and B, with a certain sequence, consider this very simple case here with a molecule which is ATCG and the complementary which is CGAT. You can write down the hybridization free energy as a sum of terms. That's intuitive. But what's perhaps slightly less intuitive is that the terms that you add up, they are not simply the contributions of each base pair. The terms that you add actually account for neighboring pairs of base pairs. So you have a contribution that accounts for this AT paired to this TA at the bottom. You have a contribution that accounts for this TC paired to this AG, a contribution that accounts for CG paired to GC. So you might ask why this is the case, and the answer is that you have this very big impact of stacking interactions. So if you are a certain nucleotide A, it doesn't matter only that you are paired with a T. It also matters what's on your left and what's on your right, because you will have stacking interactions with those other nucleotides. So to account for this strong effect of which your neighbor nucleotides are, then it is actually much more accurate to write down the free energy as a sum of terms that again account for the neighboring pairs of base pairs. And then on top of that, you have some initiation parameters that depend on which nucleotides you have at the beginning and at the end of your duplex. So in this case, AT on one side and CG on the other side. You can write down this free energy expression in a slightly more compact way. If you count, actually, you will notice that there's only 10 possible combinations of neighboring pairs of base pairs. Try to count them, and you will find that there's actually only 10, and these are listed here. So that means that for every possible DNA duplex that you have, you can write down the free energy as a sum over all of these possible combinations, so from 1 to 10, of the number of times that this particular combination appears in your sequence, times the delta G parameter for that particular combination of base pairs, plus the initiation parameters. So that's the compact way of writing this down. Of course, the delta G contribution for each of these possible combinations of neighboring pairs of base pairs, you can split in a delta H contribution and a delta S contribution. And of course, someone has measured this experimentally, doing loads of measurements of melting temperatures, typically, or calorimetry. People could calibrate these parameters, and a very famous paper in the field is this paper in PNAS 1998 by Santa Lucia, where some very good versions of these parameters are reported, and they are the ones that still we use quite often, and they are shown here. So you've got the delta H, delta S for all the possible combinations. So delta H is not very salt dependent, but delta S depends on salt. So these parameters you need to be aware of the type of salt and the concentration of salt that they've been measured at. And these are for 1 molar monovalent salt, so 1 molar NaCl. They will look different if you have divalent salt. Again, this has to do on the impact that the kind of screening has on the DNA, because DNA is negatively charged, and in particular how that impacts the entropic effects. So this gives us a very powerful tool to predict thermodynamics of nucleic acids. So once again, you can have a sequence, and from the sequence you can work out the free energy. From the free energy you can work out the stability, the melting temperature, and all that. But you wouldn't do this by hand. So there are computational tools that you can use that can help you predict how your DNA molecules are going to interact with each other, how they're going to bind to each other, how they're going to fold as a function of temperature, as a function of salt concentration, as a function of the concentration of the DNA molecules. And these tools rely on this very good understanding that we have of the thermodynamics of nucleic acids. One such tool that is very powerful is called NUPAC, and you can go and explore it. So for example, you can work out melting temperatures, free energy. So this is a much more refined tool that, the simple picture I provided. It doesn't assume the two-state model. It will tell you, if you have a bunch of DNA molecules, what the individual nucleotides are doing, if they bind to each other or they don't bind to each other. Another very important point to make is about bond selectivity. So I would like to basically make the point of how selective these DNA interactions are and how important this is for nanotechnology. So we mentioned that base pairing is selective, so A will bind to T and C will bind to G. But let's put numbers around this selectivity. So let's consider a DNA oligo of a typical length that you would have in your DNA nanostructures, say 12 base pairs. So let's consider a duplex of 12 base pairs made of 2 DNA molecules that are complementary to each other. I used some online tools to work out that if you have this temperature, so room temperature, 1 micromolar concentration of these 2 complementary DNA molecules, you have a hybridization free energy of about minus 17 kilocalories per mole, and you have a melting temperature of 53 degrees. Then I consider 2 molecules which are not complementary, they're just random, but they are still 12 base pairs. And when I did that, I got a much, much smaller, much less negative free energy, and a melting temperature which is now minus 30 degrees C. So there's a huge, huge difference in stability between DNA molecules that are complementary and the ones that are not complementary. So that's what it means for these interactions to be selective. They are really, really very selective. The other very important point is about the number of possible sequences that you can have with a certain DNA molecule of a certain length. So if you have n mer, so DNA molecule with n nucleotides, because you have 4 possible nucleotides at each location, you can have 4 to the n possible sequences. So for our 12 mer, that's about 17 million different sequences, if I did the maths right. So there's very many sequences that you can have. And it turns out that for most of these sequences, the binding free energy between them is very, very small. So they won't interact very strongly at all. And only a very small number of this very large sequence space will interact with each other quite strongly. So what this means is that you can always define a pretty much infinite, for every practical purpose, array of orthogonal interactions in a system of DNA molecules. So you can always design a DNA molecule that you call A that will only bind to B, but that will not bind to C at all. And then C will only bind to D and will not bind to F at all. And you can have pretty much an infinite number of these, again, for all practical purposes. And that's super useful because that means that you can build these complex materials and nanostructures in which you know exactly where each DNA molecule is going to go and to what these DNA molecules are going to bind. Very, very quickly, I wanted to touch on the kinetics of DNA interactions because that's also quite important. So if you go back to our chemical equation of DNA molecule A binding to DNA molecule B, you can define a binding rate that you call K on, that's a second order kind of rate constant, and you can define an unbinding rate, which is the K off. And you can link this to the equilibrium constant the usual way. So K on divided by K off is equal to the equilibrium constant. Now it turns out that K on, it's always kind of a constant. It doesn't change very much. It's always between 10 to the 4 and 10 to the 6 per second per molar. Again, it's a second order rate constant. It changes a bit if your DNA is longer, shorter, if you have more salt, less salt. It can change quite a bit if your DNA has a strong secondary structure, so it binds to itself. But unless you have very pathological cases, it's always between 10 to the 4 to 10 to the 6. If you now use this expression up here to then express K off as a function of K on, that once again is almost always the same number, then you get this exponential dependence on free energy. And that's very interesting because it means that you can change this off rate massively by changing the free energy and therefore by changing the length of your DNA. So here we put some numbers again. So if you consider a 5 nucleotide bit of DNA, you can work out an average free energy of binding between 5 nucleotide bits of DNA, and that gives you a K off of about 100 inverse seconds. So that means that the lifetime of this DNA-DNA bond, it's about 1 over 100 seconds. If you have a slightly longer bit of DNA, so 8 nucleotides, then you get a lifetime of 0.1 seconds. If you have 12 nucleotides, then the lifetime becomes 10,000 seconds. If you have 20 nucleotides, which is fairly standard in these DNA materials, then your lifetime becomes 10 to the 7 years. So you have this very dramatic change in stability of these DNA interactions, depending on the length of the bits of DNA that bind to each other. So that's useful because it means that you can make your DNA constructs very, very stable. But as we're going to see later, assuming we have time, that can also be a drawback. But fortunately, there's a way around that, which we're going to see when we talk about dynamics. So to summarize, DNA has many interesting properties from a chemical point of view and from a physical point of view that make it an excellent material for constructing things at the nanoscale. So DNA has bond selectivity. We explained what this means, that these bonds are reversible. That means that you can undo DNA bonds and make them reform if you change temperature. And this allows us to actually anneal our DNA nanostructures by weakening the interactions and then making them stronger such that they fold into the shapes that we want. DNA is a very well understood and programmable thermodynamics and kinetics. We haven't really seen this, we didn't have time, but you can have responsiveness to other types of external stimuli which are built in DNA. For example, you can have pH responsiveness, you can have responsiveness to the identity of cations that you have using non-canonical DNA nanostructures. Of course, it's biocompatible, it's very robust against degradation, or quite robust against degradation, and you can synthesize synthetic nucleic acids with any sequence, as long as they are sufficiently short, chemically, and the price of this chemical synthesis is decreasing steadily. And you can also functionalize DNA with all sorts of molecules. You can attach fluorescent molecules, quenchers, linkers, motifs to do click chemistry, hydrophobic motifs, all of that can be attached to DNA. So that's a very good set of properties, and that's why DNA is an excellent material for constructing at the nanoscale. So now let's see how we actually do that. So what the principle is of building materials and nanostructures with DNA. So that's your structural DNA nanotechnology. So after having all of this background, then it's quite easy to understand how this works. So the basic idea is that you come up with a shape or a structure that you want to build. So this can be an individual, very simple nanostructure such as this one, or it can be a more complicated material made of many nanostructures bound to each other. So you can start from either. And then you use some computational tools that rely on this very good understanding that we have of the thermodynamics of DNA. And these computational tools will help you design sequences that will then, you can synthesize chemically, you can get a company to synthesize them chemically for you, that will assemble to form your target nanostructure or your target material. So that's all that there is in DNA nanotechnology, at least in structure, DNA nanotechnology. And again, you don't have to do this by hand. There's computational tools that do this for you. Again, advertising NUPAC again, because it's a very good one. It has a design tool in which you can specify the shape of the DNA construct that you want to make. For example, this could be a stick figure, and then you tell it, I want my stick figure to be stable at 23 degrees Celsius, 1 molar sodium concentration, and 1 micromolar concentration of my strands. And this tool will tell you the sequences of the DNA molecules that will fold into your stick figure. So to be a bit more precise, what these tools are doing is that they are trying to get strands with a sequence that will fold into that particular target conformation at a thermodynamic equilibrium. So basically, your target structure is the thermodynamic ground state of your system under those specific conditions. And then you need to help your DNA molecules to reach this thermodynamic ground state that you do typically by doing an annealing. So you heat your system up above the melting temperature of all of the DNA molecules, then you slowly cool it down to allow your DNA molecules to bind to each other, find each other in the right way, and get to thermodynamic equilibrium and not get trapped in any metastable kinetically trapped states in the way. So depending on how complex your material or your nanostructure is, you might have to cool very, very slowly for this to work, or you might go a bit faster if you don't have many risks of kinetic traps. So going back to the history of structural DNA nanotechnology and to the original idea of Ned Simon, so what he wanted to do was to use these very, very simple DNA junctions that you can see here, made of just 4 strands, for example, or if you want more arms or less arms, you will have a different number of strands that interact with each other with these little bits of unpaired DNA that we call sticky ends. And what he wanted to do is to build a crystal out of this material. And why did he want to do that? Well, Ned Simon was a crystallographer, and he wanted to find a general way to help proteins crystallize. So before AlphaFold and before cryo-EM, to figure out the structure of proteins, people had to make them crystallize and then do X-ray crystallography. And many generations of PhD students have spent their whole PhD and years trying to crystallize proteins. Some proteins are very hard to crystallize. So what Ned Simon wanted to do is basically help them crystallizing using this DNA scaffold. So if you can make a DNA scaffold which is crystalline, and then you can attach proteins to this scaffold, then also the proteins will be in a crystal. And then you can do your X-ray crystallography to figure out the structure of proteins. So that was a great idea, and it was the idea from which the whole of the DNA nanotechnology field emerged, but it didn't work. And it didn't work because these DNA junctions that now we tend to call nanostars, they're too flexible. They don't like to form crystals because they're just too floppy. So they have too much entropy to form a crystalline solid. But they do form very interesting molecular liquids. They do form condensates, as we're going to see in a second. So this is a very nice paper, one of the first papers, maybe the first paper that looked at the biophysics of liquid-liquid phase separation of these DNA nanostars. So it's by the groups of Bellini and Shortino. And they mapped basically for these trivalent DNA nanostars and these tetravalent DNA nanostars their phase diagram. So this is the binodal curve of this liquid-liquid phase separating 4-armed DNA nanostars, and this is the binodal of the 3-armed DNA nanostars. So these were bulk liquids, but of course you can make them form smaller condensates if you have a lower concentration of your DNA nanostars. So again, these are tetravalent nanostars interacting with self-complementary DNA sticky ends, and they form very nice little droplets of DNA. So these are our synthetic DNA condensates. And there is an excellent review by Omar Saleh and his group that came out a couple of years ago on the biophysics of this. It's also a very good read. So in my group, we did a little bit of work on this over the years. We introduced a slight variation on the theme of these DNA nanostars by replacing these DNA sticky ends that interact through base pairing with some non-specifically interacting hydrophobic molecules, in particular cholesterol molecules. And other than this, these objects behave in a quite similar way. Perhaps the most prominent difference between these cholesterolized nanostars and the regular ones is that the cholesterolized nanostars actually can form crystals. That's because the cholesterol molecules actually bind to many cholesterol molecules. So you have an extra element of multivalency there, if you want. So if you anneal them quite slowly, these objects will form crystals, but if you anneal them fast or you change the designs in certain ways, you can make them form liquid condensates or amorphous gel-like spheres, as you can see down here. So again, I won't have time in this lecture to go through all of this, but over the years, we managed to give a lot of very interesting properties to these DNA condensates. You can control their internal structure, making them more or less porous. You can control their size, making them as big as cells or small, like nanoparticles. You can make them assemble and disassemble, subject to many stimuli. You create interesting static patterns, so you can have phase separation within the phase-separated droplets. You can create dynamic patterns, so you can have molecular reaction networks sustained within these condensates. And you can also localize within this condensate interesting enzymatic reactions to spatially control biocatalysis. Of course, Ned Simon eventually did succeed to build a DNA crystal. Just to mention, it took a long time. It took 27 years, but it did demonstrate the first example of a DNA crystal using a much smaller and more rigid and compact nanostructure called a tensegrity triangle. That's the original paper. You can read about it. So let's now move on to DNA origami, which I'm sure many of you will have heard about. So DNA origami is something that was, again, invented by one person, who's Paul Rotemund, in a very rare single-author Nature paper that you can find here. Is an amazing paper. So again, you should read it. That's Paul Rotemund. And how does DNA origami work? So it's a very, very clever idea. So what we wanted to do here, or what Paul Rotemund wanted to do here, is to make some DNA nanostructures which are a bit larger, a bit more complex than the very simple junctions or other small objects that you can make just using these short DNA molecules. And what it figured is that you can do this if you combine some short DNA molecules with a very long bit of DNA called a scaffold. So this is not a synthetic DNA. This is a DNA that comes from biology. It's the single-stranded DNA genome of a bacteriophage, which is a virus that infects bacteria, called the M13 bacteriophage. And that's about 7,000 nucleotides in length. And the idea of DNA origami is that you take this long scaffold and then you fold it in a certain shape using your shorter synthetic DNA molecules. And now, because you have this very long DNA that runs through all of your structure, that means that it's much easier to create objects which are large, more complicated than what you can do with just short DNA molecules. And of course, it's called DNA origami because you fold this scaffold into a certain shape. So again, that's the simplest thing you can do. If you look in the original paper, there's many different shapes that were already demonstrated in the first paper by Paul Rotemund. You might have seen the smiley face, which is quite famous. It's always shown by people, but you can do triangles, rectangles, square, and these are all two-dimensional structures. And then building from this, people have learned how to create three-dimensional structures of DNA origami. Again, that's a key paper on this that you can read about. And building from there, now we can build whole photonic crystals out of DNA origami. That's something that the group of Tim Liddell is working on. But a few years back, the group of Henrik Dietz could create this ginormous DNA origami assembly, which is at the gigadalton scale. And there are other techniques to create non-compact DNA origami using this wireframe design. So things that are empty rather than very compacted DNA objects. And here is where you can make your DNA people, your DNA bottles, your DNA bunnies. And also for DNA origami, obviously you wouldn't go about designing this by hand. There's quite a few different computational tools that can help you with designing your DNA origami. The original one, and perhaps still the most popular, is something called CAD Nano. And there's very many nice tutorials, for example, to learn how to use CAD Nano. that's the part of structural DNA nanotechnology done. So I don't know how much I have left, probably 10 minutes or so. We can move on to talking a little bit about dynamic DNA nanotechnology and then very briefly touching on RNA. So what about dynamic DNA nanotechnology? So here the idea is that you want to take your DNA nanostructures or DNA motifs or DNA materials and help them reconfigure or change shape or bind and unbind or do dynamic things. That's basically what dynamic DNA nanotechnology is about. And you want this to happen in response to stimuli that can be of any kind. They can be the presence of a certain DNA molecule or RNA molecule, the presence of a certain other biomolecule, could be light, could be changes in temperature, pH, and so on. And this, of course, has many applications if you want to use your DNA nanostructures for sensing, for diagnostics, for molecular computing. So what's critical for dynamic DNA nanotechnology? Again, this is a very big topic. So thinking about all of the ways in which you can actuate your DNA nanostructures will probably take a lecture series. So we're just going to focus on the most elementary, but also perhaps the most common way of doing this. And the most straightforward way to have your DNA nanostructures reconfigure is for hybridization links to form or break. So you can have two DNA molecules that bind and fall apart. And when they do this, your DNA origami might open and close. Your DNA condensate might assemble and disassemble. So that's the simplest thing that you can do. So what we want to do is basically control the kinetics of binding and unbinding of DNA molecules. So going back to a few minutes ago, what we saw is that the on rate of your DNA molecules you can't really control very well, or it's not that straightforward to do. It's always kind of 10 to the 4, 10 to the 6 per second per molar. And your off rate depends very strongly, exponentially, on the hybridization free energy of your DNA molecules. So that means that if your DNA molecules are long, they will come off very, very slowly or not at all. If they are short, they will come off very quickly. So that's undesirable because what that means is that you have a completely unbreakable link between the thermodynamic stability of your DNA system and the kinetics. If you want something that is stable, then it's not going to fall apart, obviously. And if you want something that unbinds quite easily, then it's not going to be thermodynamically stable. So that's a bit of a problem. So how do we solve this problem? Fortunately, you can solve it with a very clever trick, which is called Toehold-mediated strand displacement, which is perhaps the most fundamental operation that you have in dynamic DNA nanotechnology. So again, this is a very interesting and slightly complicated topic. We only going to give a very quick overview, but there's an excellent review that tells you everything that you need to know about this by Fritz Simmel from a few years ago, and you can read about it. So the objective here is to, again, do the most elementary operation, which is unbind a DNA duplex and form another DNA duplex. So that's what we want to achieve. And if you look at panel A here, you have a DNA duplex that we call S, formed by two molecules. And then you want to remove one of these molecules. You can call this molecule that I'm pointing at Y. And you want to replace this molecule Y with another molecule X, forming another duplex that here is called L. So normally, if you now ignore B, C, D, and E and only look at A and F, if you want to achieve that, you first need to unbind the molecule Y and then bind the molecule X. But if the molecule Y is, say, 20 nucleotides, that is never going to happen thermally. Again, it's going to take 10 to the 7 years. So it's never going to happen spontaneously. But there is a trick that you can do. So you can have this extra domain called H star, and then you have a complementary domain called H on the molecule X. And this is what you call a toehold. Either of them you can call a toehold. And what this does is that basically colocalizes the molecule X with the duplex S, such that the molecule X can displace the molecule Y one nucleotide at a time through a process called branch migration. So X can displace Y without ever having to completely detach Y. Okay? So if you switch one nucleotide at a time, you basically don't have to go over this massive free energy barrier. You cannot go around it. So again, you can think of this to climb a free energy mountain for this switch between bonds to happen. But I have a simple trick, which is if I switch one nucleotide at a time, then I don't have to go around the mountain. I don't have to go on top. I just go around in my hypothetical free energy landscape. And this is what can then catalyze this reaction effectively and make it much, much faster. So that's the trick. You can look at this in a slightly more quantitative way. Again, we won't go into detail here, but you can define an effective rate of strand displacement that tells you basically how long it takes to go from your initial state to your final state without considering all of the individual steps. And this effective rate of strand displacement, you can write it down in terms of the free energy contributions of the various steps. And what's important here is the binding rate of your toehold. So that's the binding rate of H to H star, and the unbinding rate of H from H star, K off, and then you have another rate called the branch migration rate. Maybe I should speed up a little bit. I wanted to tell you a bit more, but I'm running out of time. So read about this in this review, but the only point that I wanted to make is that with this toehold mediated strand displacement, it turns out that by just changing the length of the toehold, so changing the length of this H domain or H star domain, you can change the rate of this reaction happening, of this switching between two different DNA duplexes happening by over 6 orders of magnitude. Okay, and you can change this again by just changing how long the toehold is. You don't have to change how long this duplex X is, or how long the duplex, or how long the strand Y is. You just change H and H star. So that means that this molecule Y can be 10 nucleotides or 20 or 30 or 40. That's not going to affect the reaction rate too much. And that means that you can decouple now the thermodynamic stability of your duplex from the kinetics of the switching of the bonds. So that's the really key result. And if you look at the maths, you will see why that's the case. This has been used to actuate DNA nanostructures. So the first example of a DNA nanostructure that could be opened and closed or reconfigured is this DNA tweezers. That is also the first demonstration of this toehold mediated strand displacement. Again, very key paper in DNA nanotechnology. And this has been used to reconfigure DNA origami in a few different ways. That's a very nice result of this quite complex DNA origami Bennett linkage from the group of Carlos Castro in Ohio State University. So very, very quickly on RNA nanotechnology and RNA condensates, really it's just a couple of slides on that. So again, maybe not enough time to go through the chemistry of DNA, of RNA, but RNA is quite similar to DNA, slightly chemically different. The sugar is different. It has a OH group that the DNA doesn't have. So this makes this molecule a bit more flexible and a bit more delicate from a chemical point of view. It can self-hydrolyze quite easily. It's more flexible. And of course, you have uracil and you don't have adenine. And how does RNA nanotechnology work? Again, this would probably require a couple of lectures just to discuss this. But let's say that it's quite similar to DNA nanotechnology, but with the key difference that quite often when we do RNA, we think about folding our RNA nanostructures while they are transcribed. So you would encode your RNA construct into a DNA molecule, and then you would have RNA polymerase that reads your DNA template, and as it does, it will produce your RNA, and then while the RNA is being produced, it will fold into your target nanostructure. So this is very powerful because it means that you can now express your RNA nanostructures, for example, within cells or within synthetic cells. Many people are interested in doing this, but it also makes the design of these structures a bit more complicated. You can't anneal your RNA nanostructures if you want them to fold co-transcriptionally within your cells or your synthetic cells. So the way in which you design them and the way in which you design them to interact with each other is a little bit different. And just to finish, you can use this to make condensates. You can design RNA versions of your DNA nanostars that will fold while they are transcribed from a single RNA molecule. They will interact, instead of having the sticky ends, through these loops of RNA that we call kissing loops. They can also be complementary to each other or self-complementary. You can encode this in a DNA template. You can in vitro express this, and you will form lots of condensates when you add your RNA polymerase. You can have another version of this, which is a different color because it's a different fluorescent motif attached to it, and will also form condensates. You can express them within synthetic cells. These are simple water in oil droplets expressing your RNA condensates. You can look at them slightly more detailed, and you can make them within cells. That's a very recent result. You can read about it on bioRxiv. You can express synthetic RNA condensates within live E. coli. And that was it. Sorry for taking a bit more than allocated time, and I'm happy to take any questions.